Usually when we think about borders, whether it's like a national border or a microscopic one inside the human body, we tend to picture this static, passive wall.
Right, like a barricade.
Exactly, just a rigid barricade standing there, a bunch of concrete blocks trying to keep the bad guys out. But I mean, when you look into the reality of human mucosal barriers, specifically the vaginal mucosal barrier, that concrete wall starts looking a lot more like a bustling, incredibly complex, high-security checkpoint.
Absolutely. It's highly dynamic.
Yeah, it's constantly interrogating the environment, and it is just ruthlessly selective about what it lets through. So for you listening today, we are unpacking a stack of clinical studies and research papers focused on decoding the microscopic language of this barrier.
And we really are looking at a fundamental paradigm shift here in gynecology and regenerative medicine.
A huge one.
Right, because for decades, the gold standard for treating intimate health issues, particularly the tissue breakdown that happens as women age, has basically relied on synthetic hormones or broad-spectrum antibiotics.
Just throwing chemical sledgehammers at the problem.
Exactly, giant chemical sledgehammers. But the research we're analyzing in this deep dive moves away from those sledgehammers.
Yeah.
It focuses on how we can use the body's own natural communication network.
Yeah.
We are looking at nano-sized cellular messengers called exosomes.
Which are fascinating.
They are. And we're looking at how these invisible packages of biological code might hold the key to, completely reversing tissue atrophy, fighting off dysbiosis, and really rebuilding the barrier from the inside out.
Okay, let's unpack this, because before we get into the wild, almost sci-fi aspects of these cellular messengers, let's look at the architecture of the barrier itself. To understand how exosomes rebuild the tissue, you really have to know what's physically breaking down in the first place.
Right, you need to know the anatomy of it.
Yeah. So I like to picture the vaginal mucosal barrier as a brick fortress wall. The epithelial cells, the physical cells lining the tissue, those are your bricks.
Which are sturdy and they stack together well.
Right, but if you just dry stack bricks, if you don't use anything between them, water and microscopic invaders can slip right through those tiny gaps. So the actual security of the wall relies entirely on the mortar sealing those spaces.
And if we dive into the physical hardware mentioned in the sources,
Yeah.
that biological mortar is composed of highly specialized structures called tight junction proteins.
A mortar.
Yes, because you can't just have cells sitting next to each other, right? They have to be physically fused. The research highlights three major structural proteins doing this heavy lifting. There's zonula Occludin-1, which the literature usually just abbreviates as ZO1.
ZO1?
And then there's Occludin and Claudin-1.
ZO1, Occludin, and Claudin-1. So how do these three actually, work together to create a waterproof seal? Because I'm imagining them acting somewhat like a biological zipper between the cells.
A zipper is actually a really highly accurate way to think about it. So inside the cell itself, you have ZO1. ZO1 acts as the intracellular anchor. It binds tightly to the cell's internal skeleton, the actin cytoskeleton, so it forms the base.
Got it. So ZO1 is inside, holding on tight.
Exactly. Then, protruding out from the cell membrane, out into that empty space between the cells, you have Occludin and Claudin-1.
The zipper teeth.
Yes, these transmembrane proteins reach across the gap and literally interlock with the Occludin and Claudin-1 proteins reaching out from the neighboring cell.
That is wild.
It is. They zip together so tightly that they basically eliminate the intercellular space entirely. They form this impermeable seal that stops pathogens, toxins, and even water from leaking through the tissue lining.
Wow. So if ZO1 is the anchor and Claudin-1 and Occludin are the interlocking zipper teeth, what actually controls their production? Because looking at the studies involving the overactimized rat models.
Right. The standard way researchers simulate a sudden menopausal estrogen crash.
Yeah, exactly. The moment that systemic estrogen drops in those rats, the entire fortress wall just collapses. Why is that?
Well, the cellular mechanism behind that collapse is fascinating, and it just highlights how dependent this barrier is on endocrine signaling. I mean, estrogen acts as the site manager for this whole construction project.
The site manager, I like that.
Right, so it binds to receptors on the epithelial cells and constantly signals them like, hey, transcribe the genes for ZO1, Occludin, and Claudin-1, keep making the mortar.
But when you remove the manager...
The workers stop working.
Mm.
When you remove that estrogen signal, the cells simply stop manufacturing the mortar, the existing tight junctions degrade, and they just aren't replaced.
Mm.
The Claudin-1 zippers unlock, the ZO1 anchors detach.
So the bricks literally just start drifting apart.
They do. The tissue physically thins out, it loses its structural elasticity, and it atrophies. the barrier becomes highly porous.
Which is terrifying when you think about what's trying to get in.
Oh, absolutely.
Yeah.
And this microscopic unlocking, this loss of the Claudin-1 and Occludin seals, is the direct cause of the macroscopic symptoms millions of postmenopausal women experience.
You mean like the severe vaginal dryness, burning, thinning tissue?
Yes, all of that, plus a massively heightened vulnerability to chronic infections, because the wall is literally open.
Okay, but I have to push back here for a second. Because the exact same sources show that when researchers gave those estrogen-depleted rats hormone replacement therapy, the ZO1 and Claudin-1 proteins came rushing back, right?
We did, yes. The tight junctions restored themselves perfectly.
So if we already have a functional fix, I mean, why are scientists spending millions of dollars looking for another way to rebuild the wall? Why not just prescribe estrogen and, call it a day?
Well, if we connect that to the bigger picture, you have to look at the clinical reality of hormone replacement therapy. HRT is incredibly effective for many women, but it is absolutely contraindicated for others.
Oh, right, like with certain medical histories.
Exactly. You have patients with a history of estrogen receptor-positive breast cancers, ovarian cancers, or severe cardiovascular risks, like deep vein thrombosis.
Yeah, you can't just give them systemic hormones.
Right. No, you can't. For a breast cancer survivor, introducing estrogen back into the system, even locally sometimes, can carry unacceptable risks of triggering a recurrence. And furthermore, some patients simply do not tolerate the side effects of hormone therapies.
So they're just left without a solution.
Right, and this creates a massive clinical void. Millions of women need a way to tell their cells to manufacture those Claudin-1 and Occludin tight junction proteins, but we cannot use the estrogen signal to do it. We need an alternative biological command.
Which brings us to the actual environment outside that wall, because it isn't just the host's hormones controlling this real estate.
Not at all.
The vaginal environment is a massive, complex ecosystem of bacteria, the vaginal microbiome. And these bacteria, they aren't just passively hanging out. They are actively broadcasting signals that dictate whether that fortress wall stays strong or gets dismantled.
The microbiome is arguably the most active participant in local tissue homeostasis. And this is exactly where the concept of exosomes enters the narrative.
Okay, let's set the physical scene for you listening. Imagine that outside our brick fortress wall, there is a deep, highly protective moat.
The mucus moat.
Right. In the human body, this moat is the cervicovaginal mucus. But it's not just water, is it? The studies describe it as a thick, complex network of glycoproteins called mucins. I kind of imagine it functioning somewhat like a dense, sticky biological spider web.
A negatively charged biological spider web, to be precise. The mucus barrier employs two main mechanisms to trap invaders. There are steric traps and electrostatic traps.
Steric meaning physical size, right?
Exactly. Steric hindrance simply means the physical gaps in the glycoprotein mesh are incredibly small, so large things physically get stuck, like a big fish in a small net.
And the electrostatic traps.
Those rely on the fact that the mucins carry a negative charge. This brinds and neutralizes many pathogens based purely on their surface chemistry. So whole bacteria, whether they are beneficial or pathogenic, are generally much too large to swim through this moat. They're just trapped in the outer layers.
Okay, so if the bacteria are trapped way out in the moat, and the epithelial cells are safely behind the fortress wall, how are they communicating? I mean, they can't just shout across the gap.
No, they have to send messages across the moat. And they do this by sending out bacterial extracellular vesicles, or BEVs.
BEVs, which are essentially bacterial exosomes, right? Like messages in bottles.
Exactly like messages in bottles. Yeah. They are these tiny spherical lipid bilayers that the bacteria pinch off from their own membranes, and they pack them with a payload of RNA, proteins, and metabolites.
And they're small enough to get through the net.
Yes. The physics of these BEVs are what allow them to bypass the host's defenses. The sources detail the astonishing scale we are talking about here. For example, the vesicles released by Gardnerella vaginalis are around 134 nanometers in diameter.
Wow. Just to put 134 nanometers in perspective for you, that is thousands of times smaller than a single human hair.
It's unimaginably small. And because of this extreme nanosize, they completely bypass the steric traps of the mucus net. They just slip right through the holes.
And they don't get stuck to the negative charge either?
Right, because they have specific surface properties.
Yeah.
They don't get caught in the electrostatic traps. They diffuse freely through the thick mucus and make direct physical contact with the vaginal epithelial cells.
They bypass the security checkpoint completely.
They do.
Okay, here's where it gets really interesting. because these little lipid envelopes are loaded with instructions, but the nature of those instructions entirely depends on the species sending them.
It's a microscopic turf war.
Yeah, the research paints a really vivid picture of microbial warfare. It's a battle of good versus bad. Let's start with the good guys.
So in a healthy vaginal microbiome, the dominant species are predominantly Lactobacillus. The sources heavily feature Lactobacillus crispatus.
The ultimate good guy.
Right. When this beneficial bacteria thrives, it constantly sheds BEVs that cross the mucus and deliver highly protective payloads to the host cells.
Like care packages.
Exactly. The research shows these specific vesicles actively promote wound healing in the epithelium. But even more remarkably, they contain specific bacterial proteins and metabolites that actively inhibit viral infections.
Wait, really? Like, they block viruses?
Yes. The studies demonstrated that Lactobacillus BEVs physically block the attachment and entry of the HIV-1 virus into human immune cells in localized tissue models.
That is incredible. They are essentially sending microscopic care packages to fortify the human host because they want their home to stay healthy.
Precisely. They have a vested interest in the wall staying strong.
But on the flip side of that coin, we have the dysbiotic or harmful bacteria.
Yes. The primary culprits highlighted in the research are Gardnerella vaginalis and Mobiluncus mulieris.
And what happens when they take over?
When the microbiome loses its healthy balance and these dysbiotic strains dominate, they start pumping out their own BEVs. And unfortunately, the human vaginal epithelial cells internalize these pathogenic messengers very efficiently.
And when the human host cell opens up that message from Gardnerella, it's not a care package, is it?
No, it's a biological bomb.
Yikes.
It's an explosive inflammatory trigger.
Yeah.
The data shows that when these dysbiotic BEVs enter the host cell, their payload forces the cell to massively upregulate the production of interleukin-6, or IL-6.
IL-6, which is a major inflammatory marker.
Right. It's a potent inflammatory cytokine. It acts as a severe distress signal, sending the local immune system into absolute overdrive.
But wait, hang on a second. Isn't inflammation supposed to be a protective response? Like, why is the immune system going into overdrive a bad thing for the wall?
Well, acute inflammation is protective, yes. But chronic, localized inflammation, which is what these bacterial exosomes trigger, is highly destructive to tissue architecture.
Oh, so...
The IL-6 cascade signals local white blood cells to release enzymes called matrix metalloproteinase.
Or MMPs.
MMPs.
Oh.
You can think of these enzymes as molecular scissors. They literally start cutting up the extracellular matrix. And crucially, they actively chew apart the ZO1, Occludin, and Claudin-1 tight junction proteins.
Oh, wow. So the inflammation triggered by a bad bacteria's exosomes literally eats the mortar out from between the bricks.
Exactly. The tight junctions dissolve, the Claudin-1 and Occludin zippers are destroyed, the barrier becomes highly permeable, and pathogens can now leak directly into the underlying tissue.
So they basically trick the body into tearing down its own wall.
That's exactly what they do. This exosome-driven destruction is the fundamental mechanism behind bacterial vaginosis. And if this highly permeable, inflamed state ascends into the reproductive tract, the sources link it strongly to severe obstetric complications.
Like preterm birth, right?
Yes, premature rupture of membranes and preterm birth. The pathogenic bacteria never even had to touch the host cells. They just delivered the biological blueprints to make the host cells destroy their own tight junctions.
That is deeply unsettling, but also incredibly illuminating. We have this delicate system where the wall breaks down either through aging, when the estrogen site manager retires and stops ordering Claudin-1 and Occludin, or through dysbiosis, where bad bacterial exosomes trick the body into tearing down its own mortar with molecular scissors.
Right. Those are the two pathways to collapse.
But the underlying truth across both of those scenarios is that exosomes are the native, recognized communication network of this environment, which leads us to the ultimate question of this deep dive. If human epithelial cells are already hardwired to receive these nano-sized messages and execute their instructions, can regenerative medicine hijack that communication system?
And that is exactly what the latest regenerative studies are proving is possible.
Okay, this is the exciting part.
It really is.
Yeah.
If we need to regenerate postmenopausal tissue without relying on estrogen, we have to deliver therapeutic tissue-building instructions directly into the cells. To do this, researchers are turning to mesenchymal stem cell exosomes, or MSC exosomes.
Stem cell exosomes.
Yes. Specifically, the literature highlights exosomes isolated from human umbilical cords, called UCMSCs, and exosomes derived from adipose, or fat tissue, known as ADMSCs.
Wait, I need to pause here. If lactobacillus exosomes are already known to be beneficial and protective, why are scientists pivoting to human umbilical cords and fat cells? I mean, why not just harvest the good bacteria's exosomes and use those to heal the tissue?
Well, it comes down to the difference between defense and structural regeneration.
Oh, okay.
Lactobacillus exosomes are fantastic at modulating the immune system and blocking pathogens. But a bacterial cell cannot give a human cell the precise genetic blueprints to build complex human structures.
Right, because they're bacteria, not humans.
Exactly. They can't tell a human cell how to build blood vessels or thick stratified squamous epithelium. For true structural regeneration, you need human-derived signals.
That makes perfect sense.
Mesenchymal stem cells are the body's master builders. Their exosomes are explicitly packed with the mRNA and growth factors required to tell aging human cells to divide, synthesize collagen, and rebuild complex tissue architecture.
So let's look at how they actually execute this in the lab. In the pivotal study looking at those ovary-ectomized, estrogen-depleted rats, the ones with severe vaginal atrophy, how did they extract these master builder exosomes and actually get them into the tissue?
Well, the extraction is a feat of bioengineering in itself. To isolate pure exosomes from umbilical cord stem cells, researchers use a process called high-speed centrifugation.
Basically spinning it super fast.
Exactly. They spin the stem cell culture fluid at incredibly high speeds, generating immense g-forces. This process separates the microscopic particles entirely by density, allowing them to extract a purified, highly concentrated liquid of just the nano-sized exosomes.
But I mean, you can't just squirt liquid exosomes into the vaginal canal. The body would just flush them away before they could enter the cells, right?
Precisely. They need a highly sophisticated delivery vehicle, a structural scaffold. So they load these purified exosomes into a hyaluronic acid hydrogel.
Wait, hyaluronic acid, like the ingredient in my daily face serum?
Yes, the very same.
How does a cosmetic moisturizer act as a scaffolding for stem cell messengers?
Well, it's actually the perfect biological material. Hyaluronic acid is a naturally occurring sugar molecule in the human body. Its defining characteristic is that it can bind up to 1,000 times its weight in water.
Which is why it's in all the serums.
Right. And when engineered into a hydrogel, it forms a three-dimensional cross-linked lattice structure. It's essentially a highly breathable, moisture-rich sponge.
Okay, I can picture that.
This lattice traps the exosomes, protecting them from immediate degradation and acting as a slow-release mechanism. The hydrogel physically adheres to the atrophied vaginal wall, hydrating the dry tissue while steadily releasing the stem cell exosomes directly into the epithelial cells over time.
So just to give you the visual here, instead of a synthetic estrogen cream, they applied a slow-release, hydrating sponge completely packed with stem cell blueprints telling the tissue to regenerate.
Exactly.
And looking at the physical results on the tissue in these studies, I mean, the data is staggering.
The regenerative results were profound. The hydrogel significantly promoted the proliferation of the human vaginal epithelial cells. When researchers analyzed the treated tissue, they looked for specific biological markers. They found a massive upregulation in Ki-67.
Ki-67 being the universal biological flag that tells us cells are actively dividing and multiplying, meaning the tissue is physically rebuilding its thickness.
Correct. But even more critically for atrophy tissue, they saw a massive increase in CD31 staining.
What does CD31 indicate?
CD31 is a primary marker for angiogenesis. The exosomes delivered high levels of VEGF, which is vascular endothelial growth factor.
Let's explain VEGF, because that's crucial. VEGF acts kind of like a biological flare gun. It signals the local endothelial cells to start dividing and constructing brand new microscopic vascular highways, branching out like tree roots to bring fresh blood supply to the area.
And that new blood supply brings oxygen, nutrients, and natural systemic hydration back to the starved mucosal tissue.
Wow.
And it doesn't stop there. Furthermore, the stem cell exosomes actively disarm the oxidative stress that was accelerating the aging process in the first place.
How so?
They delivered potent antioxidant enzymes directly into the cells, specifically superoxide dismutase, or SOD. SOD acts like a chemical sponge, neutralizing the free radicals that literally tear apart cellular DNA.
But we have to bring this back to our primary focus for today. We started this whole discussion talking about the fortress wall, the tight junctions, the biological zipper. Did the stem cell exosomes actually rebuild the mortar? Did they fix Claudin-1 and Occludin?
Yes, they did. And the mechanism of how they did it is really the key to this entire field of regenerative medicine.
Okay, bring it down for us.
When the MSC exosomes merged with the depleted epithelial cells, they didn't just generally tell the cells to grow, they literally handed them the missing blueprints. The exosomes dumped specific microRNAs into the host cells.
So they gave them the instruction manual.
Exactly. These RNAs interacted directly with the host's ribosomes, the cell's protein factories, and commanded them to synthesize fresh Claudin-1 and Occludin proteins.
Oh my gosh. So the cells began manufacturing their own mortar again.
Yes. The Claudin-1 and Occludin zippers reached out across the intercellular space, locked together, and completely restored the impermeable integrity of the mucosal barrier.
They completely rebuilt the wall. And they did all of this, the cell division, the new blood vessels with VEGF, neutralizing the free radicals, and synthesizing fresh Claudin-1 and Occludin tight junctions. They did all of it without ever introducing a single drop of systemic synthetic hormones into the patient.
Which is exactly why this is such a monumental leap forward for clinical practice. For the millions of women dealing with the painful daily realities of postmenopausal vaginal atrophy, the severe laxity, the chronic dryness, the dyspareunia, which is pain during intercourse.
Right, it's debilitating for so many.
It is. And this represents an entirely new therapeutic pathway. It offers profound structural regeneration for patients who cannot, or choose not to, use hormone therapies.
We've covered a massive amount of microscopic ground today. So what does this all mean for the future of medicine? Let's zoom out and look at the journey we just took.
It's quite a journey.
It is. We started at the fortress wall. We broke down the mechanics of the tight junctions, the Claudin-1, the Occludin, the ZO1, and how they act as the biological zipper that seals the vaginal barrier.
Right.
Then we waded into the glycoprotein mucus moat, exploring how nano-sized bacterial exosomes navigate those steric traps to either deliver protective care packages, like our lactobacillus friends, or trigger highly destructive IL-6 inflammatory bombs that literally chew the Claudin-1 wall apart with molecular scissors.
The microbial warfare.
Exactly. And finally, we explored the absolute bleeding edge of regenerative science, where bioengineers are harvesting umbilical stem cell exosomes, packaging them in hyaluronic acid hydrogels, and using them to hand aging cells the exact RNA blueprints they need to reconstruct their own tissue.
And the core takeaway for you listening is that we are witnessing a fundamental evolution in how we interact with the human body. We are moving away from chemical dominance.
No more sledgehammers.
Exactly. We are no longer just shouting at the body with broad-spectrum antibiotics or flooding the system with synthetic hormones to force a change. We are learning to speak the body's native language.
Which is exosomes.
Yes. By utilizing exosomes, we are whispering highly specific, incredibly complex instructions directly into the cellular machinery, triggering the body's innate capacity for profound self-healing.
Moving from shouting with chemicals to whispering with exosomes, it is a brilliant way to conceptualize the future of medicine. And that leaves us with a final, mind-expanding question to ponder long after this deep dive ends.
I love these. Let's hear it.
If we can successfully engineer a hyaluronic hydrogel filled with human stem cell exosomes to physically reverse tissue atrophy, build new blood vessels, and rebuild the Claudin-1 tight junction barrier.
Which we know we can.
Right. And we also know from the microbiome research that beneficial lactobacillus exosomes naturally protect human cells against viruses like HIV-1 and neutralize harmful bacteria. What happens when we combine them?
Oh, wow.
Could the future of gynecology be a highly customizable, dual-action exosome gel? One that not only rebuilds your vaginal wall from the ground up using stem cell blueprints, but simultaneously programs that wall using bacterial exosomes to permanently repel recurrent UTIs and bacterial vaginosis.
That would change everything.
It really would. We could be looking at a future where a simple topical hydrogel acts as a localized living vaccine, turning a crumbling, painful barrier back into an impenetrable, perfectly guarded fortress.